Hello everyone, welcome to another video of soil microbiology. Today we're going to talk about microbial growth. First of all, let's talk about what is the constituents of a microbial cell. Most of the microbial cell matter is water, as you would imagine. But in the dry weight, you have that the 50% of the dry weight would be protein, 10 to 20% will be cell wall materials, and 10% will be lipids, and 10 to 20% could be RNA, and only 3 to 4% will be DNA. The reason that we have so much more RNA than DNA is because of the ribosomes on the cells. So that's why you have so much more nucleic acids in form of RNA than DNA. So the microbial growth means, well, when we talk about microbial growth, we are talking about the increase in number of different cells of the same microbe. One cell can split and become two cells, and these two cells can split again and become four cells, and so on and so forth, until one cell can become a whole colony. The rate of growth, the rate of division is a really important factor in this process that regulates how quick one cell can become a colony and how quick can an infection in a body progress, and also how quick can a bacteria grow in a biodigester, for example, in biotechnology, or how quick can a bacteria colonize a root, for example, or anything like that. So you have slow growers and quick growers and different behaviors in different microbes. We're going to see the phases of microbial growth in a little bit. And we're also going to talk about the factors that regulate that growth. That speed in which the microbes growth is not uniform at all in different conditions, in different environmental conditions. So we're going to talk about what conditions regulate this growth, this microbial growth. So we're going to talk about the binary fission, generation time, growth curve, and how we quantify this microbial growth. So first of all, let's talk about the binary fission. How microbes grow is by splitting the cell and becoming one cell becoming two cells. So how does that work? First of all, in one cell you have the replication of DNA, and then this cell start elongating, and this elongated cell separates these two copies of the DNA, and a septum starts to grow until the cell is completely individualized and separates from each other. So that's the process and how the binary fission works. So here's an example how this progresses in time. So one cell becomes two cells, two become four, four become eight, and then you can see that this process is an exponential progression. So the time it takes for one cell to split in two, into split in four, that's called the generation time. The time it takes for the number of cells in the culture to double. So this generation time is different for each microbe, and it's different for the same microbe under different environmental conditions. So the quickest you will get is, for example, for E. coli, it can get to 17 minutes, normally around 20 minutes in very nutrient-rich medium. But you can get in average for bacteria around 1 to 3 hours of generation time. So it's a time it takes for the number of cells to double. So because it's an exponential process, in the exponential phase of growth that we have here, then you can calculate that if you know the concentration of cells in two different times, then you can calculate that generation time by using this process that I'm going to explain here on the left. So the growth rate here is the logarithm of the concentration, the number of cells in the point B at the time 12 hours divided by the number of cells at the time B, capital letters, at 8 hours. That's divided by T, that's the growth rate. So how many cells per hour this microbe is producing. So with that growth rate, then you can calculate the generation time by dividing the logarithm of 2 by this growth rate. So at the end you get this formula here that you got time multiplied by the logarithm of 2 divided by the logarithm of the ratio between the two concentrations of cells at these different times. At the final time divided by the initial time that you're measuring. So we can solve here if you have, for example, here 10 to the 4th and 10 to the 7th cells. Then you can calculate this in 12 hours, 8 hours here for the time. So the time will be 12 minus 8, 4, times the logarithm of 2, which is 0.6931, divided by this logarithm here on the bottom that adds to 6.91. So the result of this equation is 0.4012 hours, or generation time of 24 minutes. So this is just an example of how you can calculate the generation time by having the concentration of cells in two different times during the exponential growth phase, because we're going to explain here not all the time these cells are experiencing exponential growth. So let me come back to this. So the phases that we have the exponential growth just here in this phase. But before, if we just inoculate a culture, a broth, the microbes will not experience right away the exponential growth phase. It will have first a lag phase, which will last a couple of hours, where the microbes are adjusting, adapting for the stress of being harvested and reseeded into this new broth, and adapting to the environmental conditions of this broth. But whenever they start splitting, they will quickly enter the exponential growth phase, which is this part here. And this will, of course, stop whenever the microbes sense that they have too many of them in the environment, or that the nutrients on the environment are being depleted. Then they will stop splitting and you will hit this stationary phase. And then when, of course, all the resources on this medium are depleted, you will have the death phase, which the number of microbes will decrease in time. So that, of course, depends on the type of broth that you're having and the environmental conditions that you're having, the temperature and other factors that we're gonna detail in a little bit. So how do we measure? How do we measure the cell growth, the number of cells in a specific medium? There are direct methods for estimation of microbial growth and indirect methods. The direct methods, we're gonna basically use either microscopy counting, or we're gonna do it on plates, yeah, or a cytometer, or a cell cytometer counter that we would have, like the culture counter. The indirect methods, we will have the effect of turbidity, because the more the cells grow, the more optical density you have on that medium. And we can also assess that by using some metabolic assay, like the production of CO2, for example, or the increase in concentration of any metabolite, like proteins, for example, or lipids, or DNA, for example. If you could measure that, it will be proportional of the number of cells in the medium. The other alternative is just, if you have enough cells, you can just have dry weight determinations. So let's detail a little bit more these. So for the microscopic count, you would just fix those cells in a slide and look at the microscope. And if you have some special slides that have a fixed area that you can quantify the number of cells per area and then infer how much was on the whole or per volume of liquid that you put on that chamber. When you have the cell counter, the flow cytometry, or the industry of milk industry would use, for example, the Coulter counter, which would uses just an electrical stimuli to an electrical sensor to quantify cell by cell how many cells are passing through this tube that is going through this sensor. In the standard plate count, you would just subsample the medium at specific times and plate them and see how many colonies are forming from that subsample that you've taken. Of course, you would have to have some sort of dilution to quantify this at advanced times, because otherwise you wouldn't have too many colonies to count on a plate. But that's also a standard method for assessing microbial growth. The indirect methods, you will have the Turbidimetric method, which is just submit your sample to a spectrophotometer and measure optical density, and the optical density will be proportional of the number of cells, and you can use that as an indicator, a direct indicator of cell concentration on the medium. Of course, if that's become too turbid, you can also use dilute the sample and measure again the optical density in diluted samples. You can also measure here metabolic activity. Here is a drawing here on the right, which is a setup for measuring CO2 production in a fermentation system. Let's talk a little bit about the environmental factors that regulate microbial growth. You have, among the environmental factors, you would have nutrients, temperature, atmosphere, as a way of saying gas requirements, pH, and osmotic pressure. So each organism will have a different requirement for each one of these growth factors, and some of them will tolerate a bigger or smaller range of these factors, depending on their adaptability, of course. So first of all, let's talk about nutrients. The macronutrients for microbial growth are carbon, oxygen, nitrogen, hydrogen, phosphorus, sulfur, potassium, calcium, magnesium, and iron. But we normally talk more about carbon and nitrogen specifically, and the carbon to nitrogen ratio is always something that we always look after when we look in the nutrient relationship with microbial growth. Also, phosphorus is very important, but less important than carbon and nitrogen. The micronutrients that we would have here would be zinc, copper, molybdenum, manganese, cobalt, nickel, vanadium, chloride, barium, selenium, and silica. So the other factor that regulates microbial growth is temperature. And regarding temperature, we can divide microbes for their ability of living in different temperatures. In very cold waters, we will have the psychrophiles of very cold soils. Psychrophiles would mean cold loving, and they would live between 0 and 15 degrees Celsius. The psychotrophs, they would tolerate or live especially between 20 and 30 degrees Celsius. That's normal room temperature and normal temperature for most soils. The mesophiles have a wider range. They go from 20 to 40 degrees Celsius. The thermophiles would go from 45 to 80. The thermoduric, they are thermophiles, but they can also survive for short exposures to very high temperatures, over 100 degrees sometimes. And the hyperthermophiles are the ones that are adapted for continuous survival and temperatures over 80 degrees Celsius. Most of the hyperthermophiles are archaea. In this graph here on the top, we have the relationship between the rate of growth and temperature, and then we have an optimum temperature that will maximize the rate of growth from these bacteria depending on the range that they are adapted to live in. And here on the bottom, you would have, for example, a growth curve for the same microbe at different temperatures, and having that with increasing temperature, you have increased growth for this same microbe. So the atmospheric requirements, we're talking mostly about O2 and CO2 requirements. So normally we assess this when we put a bacteria on agar medium in the test tube, and if the bacteria colonizes the bottom of this tube where it has very little oxygen, this is an anaerobe. And if it colonizes the top of the tube where you have contact with oxygen, then you would say that this microbe is an obligate aerobe. If it colonizes the whole tube, then you would say it's facultative anaerobes. And if it colonizes the whole tube also homogeneously, you would say that it's aerotolerant anaerobe. So normally you would say that the microbe is tolerant to the presence of oxygen. And why is that? Why is that the oxygen is toxic for some organisms? The reason is oxygen creates unstable oxidizing molecules like the superoxide, for example, the O2 minus, and the hydrogen peroxide, it's H2O2, which are strongly oxidizing agents. So these agents will break down, oxidize different molecules in the cell, including proteins and DNA, and therefore may cause the death of the cell. So how is it that the aerobic microbes or the facultative anaerobes deal with the presence of oxygen and this oxidizing pressure that they have? The way that they do it is by producing two enzymes mainly. There is the superoxide dismutase, which produces transform the superoxide in hydrogen peroxide, and therefore the next enzyme that's on that cycle is the catalase, which will transform the hydrogen peroxide back into O2 and water. So that's the mechanism that microbial cells and all cells use to cope with oxidative stress. So if we come back for that classification of how the microbes deal and tolerate or need different atmospheric requirements or O2 requirements, you will have the aerobes here first, the strict aerobes. They need O2 on that medium, otherwise they will not survive. You would classify them as obligate aerobes, which you would need at least 20% of O2. And you have the micro aerophile, which can survive at lower concentrations of O2. And best grow in those conditions, like Campylobacter, for example, or Helicobacter. But when you have that these cells are intolerant to presence of oxygen, then you have anaerobes. These are anaerobes or obligate anaerobes, strict or obligate anaerobes. They need to grow only when there is no oxygen on this medium. In this category, you have two subcategories. That is the obligate anaerobe that only grows in absence of O2, and you have aerotolerant anaerobe, which can survive in O2 but will not utilize O2 in the energy metabolisms. So these aerotolerants could also be classified in facultative anaerobes. Facultative anaerobes, they grow in the presence or absence of O2, but grow best under anaerobic conditions. So they prefer anaerobic conditions, but if you have some oxygen, they will tolerate that oxygen. And they use oxygen for energy, for respiration. And you would have the last category is the capnophilic organisms, which require CO2 levels, like Neisseria species, for example. So to finish this, we will talk about pH, osmotic pressure, and also some other factors. When it comes to pH, we would have that the microbes that like neutral temperatures around 7, pH 7, you would have this called neutrophiles. If they like acid conditions like pH below 4, below 4.5, then you will call them acidophiles, like the acetic acid bacteria, for example. And you will have the ones that like very alkaline conditions. You call them alkalophile, also known as alkalifile. This is very important. Normally you have most bacteria here as neutrophiles, and we have the majority of fungi tending to like more acidic conditions than the bacteria. When we talk about osmotic pressure, on the other hand, we're talking about salt concentration. The bacteria that tolerate high salt concentration, high osmotic pressure, these are called halophiles. They require hypertonic solutions, or sometimes they don't require these hypertonic solutions, but they are just able to survive on these conditions, and because they spend a lot of energy pumping salts out of these cells. And because they do that, they are able to survive on high salt concentration mediums. These are normally slow-growing microbes, and they use this tolerance to high salt concentrations to as a mechanism of colonizing medium that other cells, other bacteria will not be able to grow in. So other environmental pressures that microbes have evolved mechanisms to cope with is, for example, radiation. Some bacteria has a very high tolerance to UV radiation, and also ionizing radiation. And other bacteria are able also to withstand high hydrostatic pressure. These are called barophiles. And that's all for today. Thank you.